Everybody, today we are going to do kind of a video quiz. We've got specimens here, primary setups here to look at and decide whether they're normal flora or if they're a pathogen and we should work them up. Okay, so the first one is a nasal swab. It's on a BAP and we have some growth here. We have few different organisms here. It looks like we have about 3. And there is a concern here about possible MRSA. So we isolated the staph. So if you look, there's the gray ones look like staph, could be Staph aureus. So we're worried about methicillin resistant Staphylococcus aureus. And that is what the physician wanted to find out if the patient actually had. And so the tech or the medical laboratory scientist went through and did colonize the staph and did run some susceptibility testing on that patient. I actually don't have the results for that. And but the main concern here is that, yes, we have some normal organisms on here, except for the staph-looking species. Okay, and if you look here, they worked on the yellow. Okay, so there's that yellow-ish looking organism that is staffy. Okay, the gray ones that look very similar to that would be staph also. Okay. Moving on, we're going to do a urine culture. So this one, you want to make sure to remember that if you look at this, you're streaking differently with the urine culture. Okay, you should be doing a line down the middle and then you're separating it all across to see if you are going to be able to isolate any organisms. So this is the primary setup. We have a blood agar plate and a McConkey. Nothing grew on the McConkey, but we did have a couple of organisms grow on BAP. So without the gram stain of any of these, honestly, we can't really say a whole lot. But looking at this, on here we have, let's see, like 2 different organisms, okay? And they pretty much look like normal flora. Nothing's taking over the plate like it's a pathogen, and we have nothing growing on the McConkey agar plate. Usually a UTI, a urinary tract infection, is caused by Enterobacteriaceae. It can be caused by other fecal flora, which would include a gram-positive cocci like Enterococcus. And there are other options too, like Staphylococcus saprophyticus from the vagina could go into the urinary tract of a sexually active woman and cause a UTI as well. But this basically is normal flora, and it doesn't look like we have any pathogen to work up here. So that's that one. Now we have another one. This is a peritoneal fluid. And that really shouldn't have anything grow. And look what we have here. Okay, we have a blood agar plate and a chocolate agar plate, and we have growth that matches each other. So there's one organism here, okay, and it looks really happy. This looks like a GNR because of how big those, how big the colonies are, and they kind of have what looks a little bit like an apron. It has a wavy edge that goes out. We don't see really any hemolysis there. And looking at the difference here, this is the nasal from earlier, the MRSA. You can see that even under the microscope, gram-negative rods or rods in general are larger than cocci anyway. And so here's a difference showing you what they look like on a culture plate. So the ones on the left would be gram-positive cocci, or, you know, cocci, and then the one on the right is a gram-negative rod or rod, and I'm showing you here the difference in size, okay? All right, so what do you think? If this is a peritoneal fluid and we got growth like this, a GNR growing in a peritoneal fluid, is that a pathogen or is that fine? The answer is this is a pathogen. This is Klebsiella, and that should not be a part of a peritoneal fluid. There really isn't any normal flora in a peritoneal fluid. So there could be a tear or something in the gastrointestinal tract, and that's how Klebsiella got in there. It might have been nosocomial infection from having a surgery, and maybe the instruments weren't clean, or something that touched in there wasn't clean, and then the organism flourished. I don't really know what the case was, but that was that's an interpretation right there. So the next one we're going to do is a sputum. Okay, primary setup is going to be your blood, chocolate, and McConkey. All right, so if you're looking at this, we've got blood, chocolate, and McConkey here. No growth on McConkey. Nope, sorry, there is a little bit of growth right there. You can't really see it. There it is. Something very small. Didn't really flourish. Not really much of anything to be concerned about there. But so here we have some susceptibility testing discs that are on here. We've got Uptochin as the P disc, bacitracin as the A disc. I'm not really sure what the B one is. I don't actually work at the hospital that we got this from. So this is kind of speculation as to what those are, but usually A and P are bacitracin and Uptochin. So this is a sputum. So we're worried about maybe contamination of organisms from the oral cavity on the way out. You know, the patient needs to rinse the mouth first before they can collect the sputum. Otherwise, the organisms that would come out may not flourish. And it looks like here that this is normal flora. It doesn't look like this is a pathogen. We would be looking for things like Neisseria or Streptococcus pneumoniae, and this here may be Neisseria or Haemophilus, or but I don't really think so, because it doesn't look like that on the blood agar plate, and obviously you have the same organisms on both plates. So this looks like normal oral flora. Okay, and again, the McConkey wasn't really anything to be concerned about. It didn't take over the plate. There wasn't much growth. Now I have some other ones that we're going to look at. These honestly are going to be normal flora for the rest of this video. So I want you to get used to what they look like. Okay, so this first one is a vaginal swab. Okay, this one is a vaginal swab, and we have alpha hemolytic organisms. Notice that the plate, you know, there's only really a lot of growth in the thick area, and that's really not a whole lot of growth either. This is all normal flora. You don't have anything taping over the plate, and these organisms are expected to be in the vagina. There was on the gram stain, I'm trying to remember what was on there. There were gram-positive cocci, I believe, and gram-positive rods, and that is absolutely normal. This one is vaginal swab as well. Again, normal flora. You may end up seeing some skin normal flora on vaginal swabs too, if the swab touches the skin, the labia, while it's being collected. Okay, now we're going to go with an oral location. So this is all normal oral flora. Okay, so these were taken, the oral cavity was, all these locations are swabbed purposely to get normal flora, so you can see the difference between the pathogen and normal flora. So all this growth, yeah, there's growth, but you don't need to work it up. This is all normal. Okay, same with this oral collection. Notice we've got lots of alpha hemolytic strep. That's perfectly fine. You can have anaerobes in your mouth as well, and you do, but obviously these are not anaerobic setups, so they wouldn't grow on here. These would all be facultative anaerobes. Okay, so the next one that we're doing is a rectal swab, and so the primary setup is, oh my goodness, we've got blood auger plate, McConkey, and hectonenteric auger. All right, and woo, these look fun, right? And in another situation, this might actually be considered a pathogen, like in a UTI situation, but this is actually normal flora, so this is wonderful. We have growth of probably some strep on there, maybe from skin. You know, it's very hard to collect this on yourself. It could also be enterococcus on there. We have growth on the McConkey, and that means that this is a GNR. Okay, this looks like E. coli, and it is a lactose fermenter, sorry. And that also matches the hectonenteric auger. Let's make it so you can see it, because if you look there, that is a rapid carbohydrate fermenter, and that's what the hectonenteric auger is doing, and that's what you see by that bright orange color. These are all 24-hour plates. Okay, and that's all normal flora. We're going to do another fecal normal flora, or rather rectal. And this is exciting. This is a different person. All right. And this one has a really great result as well. There's one little guy over here pretty much growing on the hectonenteric auger, and the rest of it is over here. And this totally makes sense. We've got, looks like a little bit of skin normal flora also on here. But see the McConkey auger plate? That's looking like a non-lactose fermenter, but still part of Enterobacteriaceae. We haven't done any chemical testing on this, like none of them have been tested with catalase or anything like that, so I can't really tell you more than that, but these all look very normal and wonderful. And so if you saw this in a urine, you would absolutely want to work it up, but if this is just in a rectal swab, this looks great. This looks fine. Doesn't look pathogenic. All right, starting. The next one we're going to do is a nasal swab. And so these should be normal flora as well. And it absolutely is. Okay. The point I'm trying to make with this video is just because you have growth does not mean that you have to work it up. You need to know the type of organisms that naturally live in the area of which the specimen was taken. Like blood should never have anything in it. If it does have stuff in it like this, it might actually be skin contamination because the phlebotomist didn't, or the person who collected it did not cleanse the skin correctly. Nasal and skin can be very similar. Whenever a specimen is being collected, you obviously don't want to touch anything other than the area of which you think the pathogen would reside. So the trick is knowing what's normal flora, what it looks like on a plate, and then knowing what the most common pathogens are. Okay, so the most common pathogens in a lot of places, and I'm not being specific in the statement, are usually Enterobacteriaceae and Staph aureus and Streptococcus of some kind. Those are usually the heavy hitters. Okay, I'm not going to go through every single body site and talk to you about that, but those are the main ones. But if it's normal flora in that spot and you notice that those are growing, that wouldn't be considered a pathogen. All right, so this one is still this is also a different person, a nasal swab. Notice that this is beta hemolytic, and you know that might be an issue. The person's not feeling sick though, so it should be fine. Some people actually have Staph aureus as part of their normal flora in their nasal cavity. It's about, I guess, like 25% of the population actually carries that. So that could be what this is. And that could be okay. Let's see. So that was all nasal. All right, the next one we're going to do is skin. All right, now you get to see what skin normal flora looks like. Okay, the main component of skin normal flora is Staphylococcus epidermidis. All right, it's a coagulase negative staph, and usually your staphs, you know, look nice and gray like this. So this looks just like Staph epidermidis, very possible. All right, this is on chocolate. So the skin is the blood chocolate McConkey. Nothing grew on the McConkey. Okay. All right. I don't think the other set didn't grow anything. Next one we're going to do is ear. Okay, so looking at the normal set or the primary setup for the ear, just like with the other ones, obviously too, you need to know your media. Why do we have this media in our primary setup? Well, blood is a nutritional auger that should grow just about everything. That's a facultative anaerobe. Chocolate is for those picky eaters like Neisseria and Haemophilus. And the McConkey auger is for your gram-negative rods, like in the family Enterobacteriaceae. And so those are very normal possible pathogens for this body site. So let's take a look. Really? Nothing. Okay? Again, nothing. We really didn't get anything on any of these. The people that they were collected from had just cleaned their ears the night before with hydrogen peroxide, so we weren't really expecting to get anything there. If anything, we would have probably gotten maybe like skin normal flora. We didn't really do middle ear. They were asked not to go all the way in their ear, just the opening. You know, you're not supposed to shove things down your ear. Okay, so this is a urine. Okay, so this is a urine primary setup. We have the, as you just saw before in the beginning of the video. So this one did grow. The student got overzealous and dug a little bit in the media, and that's okay. Obviously not if you're on the bench, so you would redo it if you did that in the clinical lab. But this all looks like normal skin, the normal flora. It might have some of the normal flora of the end of the urethra, but this doesn't look like anything to work up, okay? And nothing on the conchology plate. So the most common pathogens, it doesn't look like they're here. Again, depending on what the gram stain showed, you might end up working something up. But there's a couple of different types of organisms on here that usually is considered to be a non-clean catch specimen. So, you know, this person, if it had been an actual patient, didn't actually do a clean catch urine. And that's why there would be the skin normal flora. So this would be considered a contamination. Okay, so the whole point is when you're collecting from a body site, you want to make sure not to touch any of the area surrounding that you're not trying to get a culture from, because everywhere in you and on you has normal microbes that keep you healthy from the pathogens that might try to come in there and harm you and cause disease. So that's why we are doing these videos, and that's why we're doing this lab. All right, well, I hope to see you next time. Thank you so much for watching, and we'll catch you later. Bye.